All right. So thank you very much, Katie and Chris, for the invitation. I'm really pleased to be here. I'm going to take you through some work from the literature and some work from my own group on the role of early life environmental factors in shaping the gut microbiota. So I am from the University of Winnipeg. I'm part of a new DOHAD-focused research cluster called Devotion, or the Developmental Origins of Chronic Diseases in Children network in Manitoba. So this is a great fit for the work we're doing in that group. So very broadly, this is what I study, what we study in Devotion, which is how our early life, so whether that's in utero or slightly afterwards, really shapes our long-term health. And so the original DOHAD hypothesis was really around David Barker's work that we heard about already. What about our in utero environment shapes our lifelong health? But then more recently, this is sort of evolving to acknowledge that it's not just the in utero period, but maybe those first 1,000 days. And the first 1,000 days actually starts at conception, so it's pregnancy and out to the first 2 years. And so that's where postnatal exposures like breastfeeding and the development of our microbiome might be playing an important role. So I study the developmental origins of a number of chronic conditions that affect children. These are the big three, so allergies, asthma, and obesity. For the sake of time today, I'm going to focus mainly on asthma in my examples, but many of the types of evidence I'm showing you have been also replicated for allergies and obesity. So these are Canadian statistics, but I think they're similar here and in other developed countries. So 1 in 6 Canadian children is affected by asthma. It's the #1 reason why they visit an emergency room or miss school. If they're missing school, their parents are missing work. So this has large health and economic implications. And so we're interested in how these disorders develop and take root really early in life. So if we think about that, asthma or other chronic conditions, and ask ourselves, what are the important exposures in early life? These are some of the things you might think of. Some of them are maybe more obvious than others. So nutrition, of course, we all acknowledge as being important. Harmful exposures like tobacco smoking we know to be harmful, but some of the other factors are maybe not what you typically think of as your environment, so things like stress, exercise, these are all still part of our environment. And other things too that I'm interested in that we'll talk about, breastfeeding, antibiotics we've heard about a lot already today, cesarean section we'll touch on as well. And so I'm part of the CHILD study, the Canadian Healthy Infant Longitudinal Development Study, where we've thought about those mini exposures and tried to capture them all in this birth cohort. So this is a study of 3,500 families across 4 centers in Canada. You can see them down here. So we have a site in Vancouver, Edmonton, Winnipeg, this is where I am, and Toronto. And so our families were recruited during pregnancy. And then we're taking a life course approach to this study, so we're following as the children grow up. We've just completed all the 5-year assessments for the kids. We had actually a big party on Saturday to celebrate that. And we've captured very broadly the environment. So we just had a great talk on genetics. That's one thing that we're looking at, so looking at gene-environment interactions. But beyond that, we're looking at the environment very broadly. So traditional environmental factors like smoking and air pollution, but also those more non-traditional ones I mentioned like stress. We're also very interested in the socioeconomic status or environment of our families, and we're studying the microbiome as well. So these are some sort of numbers of the study. We've got lots of samples banked, stool being the most important for the microbiome work that I'll tell you about today, breast milk being my new favorite sample. I'll touch on that if I have time. And so this is a linear view of the study. We recruited between 2009 and 2012. We've had contact with the families at various time points. So at birth, we collected information from the hospital, and we got the very first fecal sample, so the meconium. That's a diaper if you can't tell. We did a home visit, so for every family in the study, our staff went to the home to do a very thorough assessment of everything from what kind of carpets the family has to what kind of cleaning chemicals they're using, how far their home is from a highway or a swimming pool. All of these factors we thought might be important in the development of asthma, allergy, and other chronic diseases. We saw the children at 12 months, at 3 years and 5 years, and we got fecal samples at the 3 and 12 months. It was deemed to be too complicated by the time they were out of diapers, and that was kind of before the microbiome became such a big and exciting field of study. So we're now planning the 8-year visit, and we're definitely going to get another sample. All right, so thinking about the early life exposures, first I'm going to focus on 3 in the perinatal period. Oops. So the first is cesarean section. We've touched on this a little bit today. these are the cesarean section rates in different countries around the world. So in some countries like Brazil, it's over 50% of babies being born by cesarean section. In the US, it's around 33%, similar in Canada, and lower in some other countries like Sweden. So certainly cesarean section is a life-saving procedure in many cases, but you can tell by these numbers, the health status of pregnant women isn't that different in Brazil compared to Sweden. So this is being chosen as an elective procedure increasingly. And so it's of interest and importance to know what, if any, effect this might be having on offspring health. Secondly is intrapartum antibiotics. So these are antibiotics given during childbirth. These are data from the CDC showing that 25% of the US population, or 1 million women annually, are exposed to antibiotics during childbirth as a prophylaxis for group B strep. So group B strep can cause a very serious infection in the offspring, and traditionally it would be standard practice, and still is in many places, to give antibiotics as a prophylaxis because we want to prevent that infection. That was on the assumption that these antibiotics had no negative impact, so why not, right? But now we're starting to question that. And then the third perinatal exposure that I'll talk about is infant feeding. So the World Health Organization recommends exclusive breastfeeding for 6 months and continued breastfeeding out to 2 years. In the US, most infants do initiate breastfeeding these days in hospital, but within the first week, 50% of them are already receiving formula, and by 6 months, less than 50% are receiving any breast milk at all. So these are the exposures that we're going to touch on. I said we'll focus on asthma as an example of a chronic disease. So are any of these things associated with asthma? I think Chris showed the exact same graph, so he gave away one of my punchlines here. So this is a meta-analysis of 23 studies showing that children born by cesarean section have on average a 20% increased risk of asthma. So these are all individual studies. A dot to the right of this center line means there's an increased risk. And you can see that in some studies the difference is not significant, but the overall pooled estimate is. So that's cesarean section. Antibiotics, a similar picture. So this is a meta-analysis of 20 studies showing a 50% increased risk of asthma following antibiotic exposure. This is during infancy. There aren't too many studies looking at the intrapartum antibiotics for asthma. But again, a clue that perhaps this exposure is important for long-term health. And then breastfeeding, we're seeing the opposite association. So this is an even larger meta-analysis, 117 studies. In this case, each dot is not a single study, but a collection of studies based on how they're defining breastfeeding. And the overall sort of pooled effect estimate here is about a 30% reduced risk of asthma in infants who are breastfed. So these are data from the child study, so my own work that was just actually published a couple of weeks ago, where we're looking at breastfeeding and asthma. This is 3 years of age, so it's not a definitive diagnosis just yet, but it is a physician diagnosis based on a clinical assessment. And so what we've done here is divide the infants into whether they were breastfed, DBM meaning direct breast milk, so fed directly at the breast milk only up to 3 months of age. And this is their rate of asthma. It's a bit light on the screen, but it's below 10%. The next bar in yellow is still exclusively breastfed infants in that they're only receiving breast milk, but they're getting some of that breast milk from a bottle. And their rate is slightly higher, and that's significant. On the top is an adjusted odds ratio that accounts for things like daycare attendance, maternal education, and potential other confounders. The next bar is infants receiving breast milk and some formula, and then the final one is the exclusively formula-fed infants. So you see this gradient that suggests that breastfeeding is protective and that it actually matters whether the milk comes from a bottle or from a breast. Anyone have an idea why that might make a difference if breast milk's in a bottle?
But the skin microbiome.
Skin microbiome, yes, we're in the microbiome mindset. Any other ideas?
It suggests that the child is in some kind of care environment.
Care environment, yeah. So we adjusted for daycare, so we're hoping that's not it, but yes. Frozen, yeah, so it could have been refrigerated or frozen, which might have an impact on some of the other things in the milk, so the bioactive components. And so these are some of and our study doesn't give us the answer yet, but yes, it could be the bioactivity of the milk. So if that pumped milk is being stored, that might be degrading some of the bioactive components, which could include microbes and other things. The milk skin microbiota, so that baby's no longer suckling at the breast, it's not getting that contact with the maternal microbes. And there are some other interesting hypotheses that I'd be happy to chat about at coffee break. But those are some of the data from our study. So moving on from the perinatal period to other elements of early life. So one is pets. That was on my big exposure slide. There's some interesting evidence, and actually there's some that goes both ways, I'll acknowledge, but showing that having a pet in the home around the time of birth is associated with risk of asthma later on. Oops. Tobacco smoke, so one that we all know is associated with harmful health effects, in particular asthma as well. And then maternal depression and anxiety. I think someone this morning talked about maternal separation. So there's been an association shown between maternal stress and anxiety during pregnancy or the early postnatal period and an increased risk of asthma. So all of these factors, some of them risk factors, so cesarean section, antibiotics, smoke and maternal stress, all associated with an increased risk of asthma, and I could show you similar data for these other conditions. And then a couple of protective factors, so breastfeeding, direct breastfeeding may be especially important, and pets. But our question, of course, is these are associations, are they causal, and if so, what's the biological mechanism? So you can probably guess, since we're here at a microbiome workshop, that one hypothesis is that these exposures are influencing the microbiome, and that is the way by which they influence the risk of asthma or other chronic conditions. So I don't need to introduce the microbiome to all of you, but these are the bugs living in us and on us, mostly in our gut, and they play many important functions to our health. So before I move on to the microbiome story, I mean, if that's the mechanism, then is there actually any evidence that the microbiome is associated with asthma? And I won't steal her thunder because she's going to tell us about it herself, but this is a paper by Mary Claire Arrieta using data from the child study showing a really interesting association between early microbiome composition and later asthma risk. So we'll hear about that later. But take my word for it, that we do think there's reason to believe the microbiome influences asthma risk. And so if that's true, then we'd like to know what is going on with the microbiome in early life. Do these factors I just described have an impact? So our microbiome changes across our lifespan, and that's what this summary figure shows. So we're born sterile, or maybe not exactly sterile. There's debates on that. But then our microbiome gets established. It changes as we get older. So it is dynamic. It can respond to changes in diet, or whether we get a course of antibiotics, or move across the world to a different environment. But we still think that the early life period is important. So it's not totally fixed like our genome, but it does get established early in life and kind of settle into a fingerprint that stays constant throughout our lifespan. So focusing on that early period then, we could ask ourselves, what are the early life factors that shape our microbiome?
That's invariant to the diet. I'm just curious. I just wonder how persistent is it? Do you have a measure of that?
How persistent is it?
And how strong, for example, is this diet by itself? I mean, that presumably would affect that.
Yeah, so diet is certainly a very major determinant of the microbiome. We see that in babies, we see it in adults too. If you shift someone's diet, their microbiome can shift quite rapidly. But we do see, and antibiotics are another example. So if you take a course of antibiotics, your microbiome is sort of obliterated or rapidly changed. But then as soon as you stop the antibiotics, it kind of goes back to more or less what it was like before. Sometimes you have some long-lasting effects, but it rebounds on an individual level back to that individual's previous state. So there does seem to be an individual component to the microbiome where we each have our own microbiomes. They can change, but they are also sort of similar across our lifespan. That's a wishy-washy answer, I acknowledge. But so we think that what happens early kind of shapes where our microbiome is going to settle into its groove for the rest of our life. So kind of the same factors that we already talked about, right, as being important and related to our later health, could feasibly be changing our microbiome. And so I won't go through all of them, but we'll look at a few. So this is a nice summary from a recent review by Tamburini, looking at the factors shaping microbiome development. So we have first in pregnancy, the mom in utero, she has her own gut microbiome that can be passed on to the infant, her own vaginal microbiome that is also going to be an exposure from the infant. Then once the infant is born, this could occur through vaginal or cesarean section delivery. That will change the types of microbes that the infant is exposed to in its entry into the world. We'll look at that. Then there are factors like antibiotics, breastfeeding, host genetics we just heard about, and the broader environment. And all of these are going to be acting on the infant microbiome as it develops in early life. And then it's still debated, but by around 1, 2, 3 years is when the microbiome settles into a sort of adult state. But those first couple of years seem to be a really critical period for microbiome development. So in the child study during my postdoc, I was part of Anita Kaczyrski's lab in Alberta. She co-leads the Synergy in Microbiota research program with James Scott in Toronto. And this is a schematic of the work we were doing. So taking those fecal samples from the child study infants at birth, 3 months and 1 year of age, doing 16S V4 sequencing on them, and then linking those microbiome sequence data to the various different exposure measures that we have, and eventually looking to link them to the health outcome measures. Now, I just mentioned the kids all turned 5, so this is a really exciting time for the study because we have our primary outcome of asthma diagnosis and a variety of others. But at the time I was doing this work, we were still waiting for these kids to grow up. So we took the first question in terms of what early life factors were shaping the microbiome. So these are not data from our lab, but this is looking at cesarean section. So this was the first sort of famous paper looking at the microbiome of infants born by cesarean section compared to those born vaginally. And so what you can see in this figure is each dot representing one sample. So in green, we have the mother's oral microbiome. In red, we have the mother's vaginal microbiome. And then in blue, we have the mother's skin microbiome. And then the lighter colored dots are the baby microbiome samples, their gut microbiome. And so the babies born vaginally are the pink ones, and you can see that their gut microbiome looks similar because it's closer in proximity to the mom's vaginal microbiome. Whereas the light blue babies are born by cesarean section, and they cluster near the mom's skin microbiome. So they were not exposed to the mom's vaginal bacteria, and therefore their microbiome is shaping up more to look like a skin. And then this is a more recent study showing, again, that babies born vaginally in blue cluster more closely together and separately from babies born by cesarean section in black. So this is looking at whole communities of gut microbiota and showing that they separate in infants based on the way they were born. So we looked at that in the child cohort. These are 2 papers that we published. The first was really a pilot study looking at just 24 infants at 4 months of age. So how did their microbiomes look depending on how they were born and also how they were fed. And then we found a difference. And so the next question was, well, if we see this difference at 4 months of age, what does that really mean? Is it a long-lasting difference, or does it resolve, and does it have any impact on health? So we looked in a follow-up paper at a larger group of 166 infants and went out to a year to see if those differences were still apparent. So this is from that follow-up study. Oops, not sure what happened there. But this is it was 198 infants. So this is at 1 year of age we looked, and we grouped the infants into how they were born, so cesarean versus vaginal delivery, and also looked at whether or not their mothers received antibiotics during childbirth. So one of the complications when you're looking at differences in the microbiome based on method of birth and trying to tease out the effects of cesarean section is that nearly all moms receiving a cesarean section also receive antibiotics as prophylaxis to prevent wound infection. So you then it's hard to say whether it was the antibiotics or the C-section itself that had this effect on the microbiome. So here, we still have that issue. So all of the cesarean deliveries in our cohort received intrapartum antibiotic prophylaxis, or IAP. But we had a group of vaginally delivered infants that also received antibiotics, usually for GBS prophylaxis. So we were able to look at the C-section babies who all got antibiotics, vaginal deliveries with no antibiotics, and then the vaginal deliveries with. And we separated out the cesarean sections based on whether they were emergency or elective procedures. And so what we're looking at here is the average microbiota profiles at 3 months of age based on the method of birth and also stratified by breastfeeding. So over here, you can see these are infants who are not exclusively breastfed, and their microbiota profiles are quite different based on whether they were vaginally delivered without antibiotics all the way to emergency cesarean section with antibiotics. One of the major differences is the depletion in Bacteroidetes, which is the orange colored phylum here. And you can see that that pattern was pretty similar regardless of whether the infants were breastfed or not. So this group has a pattern, and this group has almost the same pattern. So there's a dysbiosis after cesarean section regardless of feeding method. And there was also, importantly, kind of a gradient. So the vaginal deliveries that received antibiotics were not as perturbed as the C-section deliveries, but they were different from the vaginal deliveries. So suggesting that there's an effect, a separate effect both of the cesarean section and the antibiotics. Now, when we went out to a year, because that was our question, we see this at 3 months, but is it persistent or is it just a transient effect? This is what we saw. So over here in the breastfed babies, you can see that these profiles have pretty much evened out. So the babies born by cesarean section, by 1 year of age, their microbiome seems to be recovered. It's looking pretty much like the babies who were vaginally born. But over here, the babies who were not breastfed still have this deficit if they were born by cesarean section. So this suggests to us that babies born by cesarean section do have a difference in their microbiome. They have a dysbiosis. But if they're breastfed, this is repaired more quickly than if they are not. But I always, So this is suggesting that if a mom has to have a C-section, which we know is sometimes the case, then maybe one thing we can recommend is to be breastfed to help that microbiome recover. Does that make a difference to a health outcome? We're still waiting on those data from the child study. But in the meantime, there's this mouse study that just came out in the last month or so, where they were able to, the nice thing about the mice is you can totally separate the antibiotics from the C-section. They did C-section deliveries on these mice. They don't need to give the mice antibiotics. And what they see is a change in body mass index and also a change in the microbiome. So here we're looking at a measure of microbiome composition. And you can see in vaginally delivered infants, the normal process is that the microbiomes are different and they sort of settle out over time. But in the cesarean section delivered infants, you never see this pattern. So there seems to be sort of a deficit in the maturation of the microbiome in the infants or in the mice born by C-section. And then you can see that this has an impact on body mass and that it's sex specific. So that was something that I wanted to raise here in our discussion, is that I think when we're doing any kind of work, but also microbiome work, it's important to look at men and women or boys and girls separately, because sometimes the effects are different. So you can see here in blue the vaginally delivered mice, and then in red the cesarean delivered mice. The cesarean delivered mice have a higher weight gain, more rapid weight gain, and that's especially true in females. So a nice collection of data from both human studies and mouse studies showing that cesarean section has an impact on the microbiome and that this appears to have an impact on health outcomes.
Is there any explanation for that gender difference?
Not a great one. It's a very good question. I think in general, studies of weight gain and obesity in boys and girls do show sex differences. So boys and girls gain weight at different, and gain body mass and adiposity at different rates. So the factors that influence them may be different, but I'm not sure in this particular case if there was a hypothesis given. Does anyone in the audience know? Laura?
I can email Maria Gloria.
Yeah.
But sometimes we also know that microbes can regulate estrogens and testosterones, and so you can select for those, so there might be disrupted signaling with that might have metabolic consequences. But yeah, I'll ping Maria.
So I think some of your work has also shown sex differences.
Oh yeah, definitely. We see sex differences in general with the obesity models. We see similar trends in males and females with delayed development in the females because they don't tend to gain as much weight on a high-fat diet. But this is a different model, and I think it's very interesting that the females are showing an acceleration.
This is going back to humans now and focusing on the antibiotics. So we saw earlier the important impact of antibiotic exposure in early life on the microbiome in mouse models and in some human cohorts for obesity. So this was a Finnish cohort looking at antibiotics and looking specifically at the timing. And what overall they found, these are different periods of exposure and different types of antibiotics, so penicillins versus macrolides. And they found that there was reduced bifidobacteria in children that had been exposed to macrolide antibiotics and increased proteobacteria and Bacteroidetes, and that they showed a recovery of their microbiome by about 12 months. So just like I showed with the C-section, there was a recovery of the microbiome with breastfeeding. Here they find a recovery of the microbiome once the antibiotics are removed.
How long was the follow-up on the Finnish?
So the follow-up, they've got out to 38 months there, it looks like. I think it was different for different children, but it was definitely beyond the period of infancy that they were seeing differences, but it was the exposure during infancy that seemed to be especially important. So I always throw up this slide, and it's fun to have Laura in the room this time, but whenever I'm giving these talks, we see the microbiome recover and we think, okay, phew, as long as I breastfeed or as long as I stop the antibiotics, the microbiome recovers. But we saw from Laura's talk earlier today that that doesn't necessarily mean there hasn't been an impact on physiology that may not have a long-term impact. So we saw in her work that the antibiotics were used to disrupt the microbiome in early life, and stopping those antibiotics allowed the microbiome to recover, but there were still these lasting consequences. So this is another human study where in the child cohort we have a pretty big N, but we only have the sample at 3 months and a year. So there's a lot going on in between there. This is a really nice study where they sampled every month of life and were able to see the sort of natural course of microbiome development. And they looked at those same perinatal exposures that I've mentioned. So antibiotic exposure at the top here, method of birth, and then diet. And what you're seeing is different measures of the microbiome, so diversity, the number of observed species, and another measure of diversity. And what you can see is that antibiotics have an effect on those measures, delivery mode especially, having an effect on those measures, as well as diet, so formula versus breastfeeding. So together, I think those collection of studies show some convincing evidence that cesarean section, formula feeding, and antibiotics have an impact on the microbiome. I want to touch on a few of those other postnatal factors that I talked about earlier. So pets is a fun one. This is some work out of the child study where we looked in our pilot cohort at infants who were living in a household with a cat or a dog, and we found that those living with pets, the infants had on average higher gut microbiota diversity. So you can imagine maybe the pets bringing different microbes in from outdoors and the infants being exposed to those. And they had a different composition. So specific taxa seemed to be increased or decreased. And the Ds and Cs are dogs and cats. So we saw a difference based on what kind of pet. And so that's actually been followed up on more recently with a larger study, again, showing differences according to pet exposure. Smoking. So there's not so much evidence for this, but I tried to do a search. What I found was a study in mice showing that secondhand smoke exposure in mice was associated with changes in the gut microbiota. So you're seeing different taxa along the bottom there and different proportions based on exposure to smoke. And I also found another study looking at maternal smoking during pregnancy and then gut microbiota profiles at birth in the offspring, and some differences were seen there. And the last one was stress. So I mentioned that maternal stress has been associated with increased rates of asthma. I didn't find, but someone's welcome to correct me, any human evidence for stress associated with microbiota in infants. But what I did find was some mouse studies. So one that was very recently published showing that stress during pregnancy in mice was associated with altered maternal microbiota, as well as altered microbiome of the offspring. And again, it was in a sex-specific manner. So you're seeing there the microbiome profiles of offspring that were born to control females or females that were stressed during pregnancy. I found one other study looking at gut microbiome composition. I'm not a mouse modeler, but correlated with grid floor-induced stress. I think that's where they put mice on a floor where they can't actually step. It's like a cage, so they get very stressed. And that affects their microbiome, apparently. And then lastly, a monkey study. Maybe Katie knows this one. Looking at prenatal stress altering bacterial colonization of the gut in infant monkeys. So I left the best to last, so my favorite is breastfeeding. And certainly there's a lot of evidence that breastfeeding influences the microbiome in infants. I think someone mentioned earlier, even more so than delivery mode or almost any other exposure that you can care to look at. It makes sense, of course, the food that a baby eats is the food its microbes are going to eat. So these are data from the child cohort looking first at bifidobacteria that we've heard about already today. So classifying the infants based on their duration of breastfeeding. So never breastfed, having the lowest levels of bifidobacteria, out to 12 months or more of breastfeeding, much higher levels of bifidobacteria. Clostridium difficile, we also heard about earlier today. That was going to be my question if I'd had time for it. So we actually see lower rates of colonization with C. diff in the exclusively breastfed babies. So this is 80% of the formula-fed babies were colonized with C. diff. They don't have any symptoms, so I was curious whether we know if it's sort of a non-virulent strain or if there's something unique about the infant gut that allows them to deal with these bugs without being acutely ill. But there is evidence that babies colonized with C. diff have a higher risk of allergies later in childhood. So we do think there's sort of a clinical implication for this. And then finally, this is a measure of microbiota diversity. And so you see that it's lower in the exclusively breastfed infants. And we often talk about microbiota diversity, the more the better. Healthier adults tend to have higher diversity in their gut microbiota, but we see the reverse in babies. And it makes sense when you think about it. If babies are only receiving breast milk, that's probably selecting for a narrower but arguably optimal microbiota composition, and that's what we see there. So we've seen then these various early life risk factors for asthma and other chronic diseases do appear to be associated with the gut microbiota. Hopefully I've shown you some evidence for that this may be one of the mechanisms by which these factors are influencing the microbiome. I want to talk a little bit more about breastfeeding if I have time. So since that is such a strong determinant of the microbiome, this has been the focus of my research lately. So looking at breast milk, I like this sort of framing of milk as nature's first functional food. So functional foods are kind of a buzzword these days, and when you think about it, milk is really nature's original functional food. So these are two images that tell you the same thing, namely that there are a lot of things in breast milk that we don't find in formula. So formula, of course, has all the essential fats and carbs and vitamins and minerals that are known to be important for infant growth. But on top of that, breast milk has a whole host of interesting things, including antibodies, hormones, antiparasitics, growth factors, and enzymes that are not found in formula. And actually, this picture is kind of outdated because it doesn't include my two favorite things, which are probiotic bacteria found in breast milk and prebiotic oligosaccharides. So probiotics, of course, are live beneficial bacteria, and prebiotics are the non-digestible carbohydrates that will select for these beneficial bacteria, and both are found in human milk. So if we think of milk at the sort of quantifiable scale, we have a lot of water, then we have macro and micronutrients, and out of those, actually the third most abundant component after lactose and lipids, and even more abundant than proteins in milk, is human milk oligosaccharides. And there are a variety of different kinds, and they are food for gut bacteria. So this is a really exciting time to be studying these HMOs, or human milk oligosaccharides. These are cartoon depictions of them, so they're built of a limited number of sugar building blocks, and you can depict them in different ways. They're non-digestible to the infant, and so they've been known and discovered sort of decades ago, but for a long time no one paid much attention because we knew that infants didn't digest them. It's only now, when we realize that they're food for the gut microbes, that all of a sudden people are very interested. These are very structurally diverse, so there's only a dozen or so shown here, but we know that humans make actually more than 100 different human milk oligosaccharides. All mammals make oligosaccharides, but in different combinations and amounts. So mice, for example, make just 2 oligosaccharides in their milk. Cows make about 40, and humans, as I mentioned, make over 100. And we don't actually know a lot about why humans make so many, and we also know that each woman makes her own sort of unique mix of oligosaccharides, and we don't know much about why that is. It's highly variable. There is a genetic component to this, so a mother can be a secretor or a non-secretor. This is a genetic trait that partially determines the HMOs that she makes, but aside from that, we don't know much about this variability. There have been some small studies finding associations between oligosaccharide profiles and things like HIV transmission, risk of allergy in the offspring, but really a sort of unexplored Wild West place to be doing research right now, I would say. So what do these oligosaccharides do? Well, they're prebiotics, as I mentioned, so bifidobacteria in particular can consume them, but they have other functions as well. So they function as anti-adhesives, they bind to pathogens and prevent those pathogens from binding the gut epithelium. They are absorbed in small amounts, but they are directly absorbed into the infant's circulation, so they're found excreted in urine, meaning that they might have systemic effects. They've been shown to interact directly with some immune cells, so many potential functions. These are the HMOs that we've measured in the child cohort. Other than being pretty and colorful, I don't have too much to tell you about them today, but you can see here that they cluster in two main groups, the non-secretor mothers that do not make this red-colored HMO, which is 2-fucosyllactose, and then the secretor mothers, which is about 3/4 of our cohort, which do make that 2-fucosyllactose. So you can see there's two groups, but even within those groups, there's a lot of variation. And so we're really interested to study what is it that determines this variation, because it's, and how is that implicated in the microbiome development. So that's the prebiotics. There's also a probiotic component to human milk. So until relatively recently, it was assumed that human milk would be sterile unless the mother had some kind of infection. But we now know that that's not true. Human milk is not sterile. It actually contains live microbes, and it's been estimated that breastfed infants consume somewhere around 1 million microbes per day, which is quite a lot. And they're probably there for a good reason. So these are a source of gut bacteria. Again, there have been a few small studies, but still not a whole lot, looking at the variation in microbiota profiles. So you can see some there. Those are 3 samples per woman in 16 different women. And so you can see that even within a woman, there's variation, but there also seems to be sort of a profile that's somewhat consistent within each woman, different from others. And so at this point, we don't know a whole lot about what makes these individual profiles. And we don't know anything at all, really, so no studies have looked at the potential association of milk microbiome composition with infant health. So we are again studying this in the child cohort. These are some preliminary data. So here looking at we've done 400, so each column in this graph is one woman. And you can see, again, huge variation in the profiles of these microbiota across the cohort. On the right side, we've been looking at clustering algorithms. So in this figure, each row is a woman and each column are different types of microbiota. And you can see there's kind of an overall 3 different clusters. And we're doing work now to see if there are certain maternal traits that predict cluster membership and whether these different clusters are associated with any kind of infant health outcomes. The framework we're looking at and working with is that various maternal factors, so things like maternal age, maybe maternal diet, ethnicity, would shape her milk environment, so meaning the other macronutrients in the milk, maybe the HMOs, and that these other components in the milk would then influence the milk microbiota, and that other early life factors and infant factors might also influence the milk microbiota, because one of the ways in which the milk is thought to be colonized is actually through essentially backwash from the infant, so that it's actually infant oral microbes that are then colonizing the breast and colonizing the milk. And that all of these would then, of course, impact the infant gut microbiome. So there's been some work recently looking again at milk microbiome and trying to, through algorithms, determine whether the infant gut microbiota are actually being derived from the breast milk microbiota or potentially from the skin microbiota. So we talked about the skin exposure through the direct or indirect breastfeeding question. And so that work has been very interesting and shows that probably it's a bit of both, that the infant is consuming skin microbes but also getting milk microbes, and that's contributing to the gut colonization. So if I have 1 minute at the end, I wanted to also think about health disparities, because that was kind of what brought us all together today. And so if you think about what factors might contribute to health disparities, it's again the same list, right? So we know that socioeconomic disparities can be associated with different rates of exposure to smoking, certainly different rates of breastfeeding and other exposures. And so looking at the breastfeeding in particular, this is a map that's, I think, very telling. So this is the estimated mortality in kids under 5 attributable to suboptimal breastfeeding. So on this website, you can play with all sorts of data from the Institute for Health Metrics and Evaluation. And this shows across the world that in some countries, in particular South Africa, there's still a considerable proportion of child mortality that's attributable to suboptimal breastfeeding. So this is an area where we could really make a difference with a modifiable factor, and it's potentially linked to the microbiome. This is in the child study looking at breastfeeding inequities. So even in a developed country, we see that women who have lower education, women who are obese have lower breastfeeding rates, also particular ethnicities, younger moms, and we also see differences across different centers of the study. So certainly major differences in breastfeeding rates according to various factors. And I think there's something we could do about this. So this is I mentioned to someone earlier this morning, I hired a student from community health sciences. I have other students from sort of microbiology backgrounds. But this one came to me and she was creating some variables. She knew how to code. We had a research day coming up and I said, let's do an abstract on breastfeeding rates in the child study. She said, well, that's interesting, but I want to look at inequities. And I thought, well, how are you going to do that in our data? All she had were the breastfeeding variables, but what she looked at was breastfeeding in the first 2 days of life, so before the infant left the hospital. And so what you see in the red line is the breastfeeding rates over time, so they drop off, of course. But the red line is the babies who left the hospital exclusively breastfeeding. So after 2 days, they'd only received human milk. They breastfed on average 4 months longer than children who received formula in hospital. So I think this shows that in the first couple days, we could make a really big difference on breastfeeding rates, which could have a big difference impact on gut microbiota and long-term health. And this was especially true when we broke it up by inequity groups. So these are the lowest, moms with the lowest education, they breastfed for the shortest amount of time. These are the moms with the highest education, breastfed for the longest. And that's if they received formula in hospital. But if they didn't, this gap was really shrunk by half. So if we could help those moms with the lower education or sort of the most disadvantaged to breastfeed early, then this had really a long-term impact on their breastfeeding rates and so on and so on. So I wanted to include that from the equity
Is there any effect of
perspective.
just the duration? The child is born
Yeah.
and breastfeeding starts, say, a few days later. I mean, do we know anything about the lag? I've seen some claims that there is such a...
It can sometimes take a couple days for the milk to come in. In particular, this happens for babies who are born preterm or born by cesarean section. So these sort of, it's a triple hit to these infants. If they're born by C-section, often the mom's milk doesn't come in and the mom gets antibiotics. So it's this like triple hit on the microbiome. There are data on that, and so sometimes supplementation is going to be necessary, but there's also a lot of work being done around supplementation that's not medically indicated, and that's where you could make a difference, I think. So we can continue that discussion. That was the end, but I wanted to bring that in as an equity piece. I haven't thought much about equities in the microbiome, but this is where I thought there might be a link in early life. So maybe I shall skip these. I have a lot of unanswered questions that we can think about.
Please list them. We have time.
Okay, so what's a healthy microbiome? I think we don't know a lot about that. We see different microbiomes, but we don't know so much about, I think Emily mentioned, it doesn't maybe matter so much who's there, and that's what we've focused on a lot. It matters probably more what the functions are, but we're still sort of in the early stages of understanding that. What are the long-term effects? I've shown you associations between different early life exposures or microbiome dysbiosis, but most of this work is done in infants or in early life, and we don't necessarily know the long-term impact. I think understanding the mechanisms is important, firstly for establishing causality. So is the microbiome dysbiosis or the changes to these microbes actually causing these health outcomes? And if we understand how, then we can start to think about therapies. If there is a causal mechanism, then the first thing to do would be to prevent this dysbiosis in the first place. So thinking about early life, this might involve advocating for vaginal delivery whenever possible, breastfeeding certainly, avoiding the unnecessary use of antibiotics. If there is an unavoidable exposure to one of these things, then what can we do afterwards, right? So Laura mentioned the vaginal seeding as a really interesting potential intervention after cesarean section. While that evidence is still accumulating, I think one thing we can certainly do is advocate for breastfeeding. We have some data from child that suggests that helps the microbiome recover. Pre and probiotic approaches, fecal transplants. I think all of these are really interesting areas for future research. And then sort of the big question today, I guess, is how can we target the microbiota in early life, from my perspective as a pediatric researcher, to help reduce health disparities. So that's my image, and I'll just end off with an acknowledgement. So to the child study, this is our big birthday party on the weekend with all our kids who've turned 5, and these are all the people that helped with the work that you saw today. So thanks.
Simulation where you say, suppose now we equalize the microbiome, so we give them at least what we think is healthy. How much of the disparity would be reduced? We've just done a crude calculation.
I have not done a crude calculation, but I would love to work with you on that because I know that's your specialty. I mean, looking at the percent of variation explained by the microbiome compared to other factors,
Yeah,
for
like
example.
incidence of child disease or something.
Yeah.
Or maybe school readiness.
I mean, you saw, for example, the some of the estimates were quite big on something like asthma, 30% reduced risk of asthma from breastfeeding. I would surmise that some of that probably goes through the microbiome, probably not all of it. So I think there are effects of breastfeeding that do other things beyond modify the microbiome. But some of those could be pretty big. The C-section was 20%. Those are pretty, at the population level, large effect sizes. But whether fixing the microbiome would entirely reduce that effect, I guess that's the best case scenario.
So, last question, sorry.
Oh, yeah.
Yeah.
So this human milk oligosaccharides are very interesting. Do you have any idea? So what the mechanism is that the bacteria use to degrade this, and whether this leads to maybe diversity? Because I always have the idea that the good microbiome is diverse, but I'm not sure if that's really.
Yeah. So that's a two-part question. They're both good. Firstly, so I didn't make it very clear, but not all of the gut microbiota can consume these oligosaccharides. It's actually a select few of them. So bifidobacteria, and actually only a few strains of bifidobacteria. So bifidobacteria infantis, for example. And so I think that's one of the ways that breast milk selects for this very specific infant microbiome. And that's why we actually have a reduced diversity in the infant gut in exclusively breastfed babies, is because it's only a select few of these microbes that can digest it, and it's because they have certain genes, I'm sure, to be able to utilize them.
Thank
Thanks.
you. Thank you.